which can reduce the acetylation. And then you read this review paper and say that, okay, let's look into it. So if you look into it, if you just focus on this one, this one, 12 position, that is get acetylation. Also, you can focus here, K56, very important mark for DNA damage response. In K56, acetylation increases on glioma, colon cancer. But in this cancer, melanoma, it is decreasing. K18, pancreatic cancer, it is upregulated. In breast cancer, downregulated. What is telling to you? Specific cancer. So, a general inhibitor cannot be important for all type of cancer. So, the problem with the epigenetic drugs is to look for the specificity. If you come up with a specific enzymes, specific inhibitors, specific, then you can easily design. But don't think that same inhibitor can going to act for all. But as usual, I have problem with everything. You have grown the cancer cell. सर मैंने पैंक्रियाटिक कैंसर सेल लाइन ग्रो किया है और मैंने ब्रेस्ट कैंसर सेल लाइन ग्रो किया है, मैंने देखा कि K18 एसिटाइलेशन एक में डाउन है एक में अप है. मैं बोला आई डोंट लाइक दिस एक्सपेरिमेंट. सो you have come up with the convince me that this will be working. I'll take this. Okay. Hello. हाँ वो आएगा, बात हो गई है. 1:10 में बोला है, ठीक है. ओके. So this is something to pick up. प्रॉब्लम समझ में आ गया ना? आपने वेस्टर्न ब्लॉटिंग किया है या चिप किया है, बोला है कि सर ये फेनोटाइप है. मैं कन्विंस नहीं हूं. मुझे दूसरे तरीका से प्रूव करो कि K18 जो है वो इन्वॉल्व है. हाउ विल यू गोइंग टू डू दैट? एंड आई हैव डिस्कस्ड दिस यस्टरडे. if you have make good note or you have understood it, or this can be answered basic or biochemistry. Why I am telling biochemistry, ये सर तो पागल ही करके छोड़ देगा, खाली बायोकेमिस्ट्री सेल बायोलॉजी बोलता है और तो कोई सब्जेक्ट ही नहीं है जैसे. Because everything has been arise from there. So biochemistry, I feel personally is some other subject. If you understand biochemistry, you understand most of the thing. So उधर से कुछ निकलेगा. सो हार्मोन्स का क्या कनेक्शन है इसमें, क्या करेंगे? ठीक है, हार्मोन्स का कह रहे हो तो ठीक है, मैंने ट्रीट किया हार्मोन से कुछ हो रहा है, बट डायरेक्ट कैसे एविडेंस मिलेगा? दैट विल बी आल्सो कोरिलेटिव, राइट? एसिटाइलेशन का चिप कर रहे हो तो मार्कर है. प्रॉब्लम है कि यू ग्रोन पैंक्रियाटिक कैंसर सेल्स and you have grown breast cancer cells, and you found that K9 acetylation in one case increasing and one case is decreasing. You have measured K9 acetylation by whatever method. K9 नहीं, K18 है, sorry यहाँ पे. And I said that no, I don't believe this. आंसर यहीं पे है. यदि एक चीज समझ लिया तो बाकी चीजें तो अपने आप ही समझ जाओगे. यही मेरा पॉइंट है. बोला मेरे तरह टीचर मिल गया तो फिर तो मेरी इंटरव्यू की तो बाट लग जानी है. Yes, you have made a K18 acetylation and you write that it is increasing, decreasing. But I said that एंजाइम और सबस्ट्रेट दो चीज तो आपको पता है ना. तो एंजाइम व्हेन एक्ट्स एन अ सबस्ट्रेट, मैं एपिजेनेटिक्स सब नहीं पढ़ा रहा, तो एक प्रोडक्ट बनता है. सो आइदर यू मेजर प्रोडक्ट विच इंक्रीज, सबस्ट्रेट विल डिक्रीज. सो यहाँ पे K18 एसिटिलेशन जो है वो प्रोडक्ट है. आप मेजर करो K18 एसिटाइलेशन बढ़ रहा है, बट मैं कन्विंस नहीं हूं. मुझे क्या करना पड़ेगा? तो रीडर तो इसको, इसमें तो रीडर बढ़ जाएगा, इसमें रीडर तो कम काम करेगा. वो तो एसिटाइलेशन का रीड आउट ही देगा ना. को फैक्टर तो दोनों में एसिटाइल कोए ही है. अगर एसिटाइलेशन बढ़ रहा है तो कोफैक्टर्स का कंजम्पशन भी बढ़ जाएगा. तो वो तो एसिटाइलेशन को ही रिफ्लेक्ट करेगा ना, यदि ऐसा है तो. तो इसमें सबस्ट्रेट का कॉन्सेंट्रेशन क्या होगा आपका? वो तो बोल ही रहा है ना कि ये तो एसिटाइलेशन की वजह से, उसी चीज को आप दूसरे तरीका से बोल रहे हो. आई गॉट इट. कोई रॉन्ग आंसर नहीं है, बट मैं कन्विंस नहीं हूं. सोचो कुछ, मैं थोड़ी सी हिंट देने की चेष्टा कर रहा हूं, कोई लाउडली सोच नहीं रहा है. मान लो आपको पता करना है कि एसिटिलेशन जो है इन्वॉल्व है, तो मैं नेक्स्ट स्लाइड में चला ही जाता हूं. तब भी क्लैरिटी नहीं आएगी, लेकिन थोड़ा सा एंजाइम देख सकते हैं. एंजाइम जैसे हैट वन देख सकते हैं, या जो एंजाइम के 18 एसिटिलेशन करता है उस पर देख सकते हैं. ये एक आंसर जो आप लोग मिसिंग कर, बट फिर भी मैं संतुष्ट नहीं हूं. देयर इज समथिंग important thing missing here, and this is the problem in the epigenetics field. So without further going detail, you can measure the enzyme also, that will further support your hypothesis, the enzyme for K18 acetylation. But the best experiment will be the make mutation in the K18. यही आंसर था जो मैं उस समय से पूछ रहा था. You can make lysine to alanine mutation. You can make lysine to glutamic acid mutation using site directed mutagenesis. अभी तो आपको आंसर पता चल गया ना. I teach you, right? You can make the nucleosomes. So you can make a nucleosomes with lysine mutated to alanine. आप H3 जब प्यूरिफाई करोगे तो वाइल्ड टाइप H3 भी प्यूरिफाई करो, म्यूटेंट H3 भी प्यूरिफाई करो, दोनों का न्यूक्लियोसोम बनाओ, then you can take the direct correlation. If in that last slide, I'll put back last slide. So the last slide is basically telling you, if have a K18 mutation in the cell, it will be very easy to tell, and direct correlation can happen. So this is how you probe, right? You probe, right, that the enzymes are involved in the active side. How you do? You do site directed mutagenesis. वही तो मैं पढ़ा रहा हूं. आ गया समझ में ना? I am not teaching something. I told you I am teaching biochemistry only because you can do answer that way. Problem क्या है in the chemistry? Because there are many histones in cell line. So to make the mutation in actual cell line is not an easy task for histones because there are many genes which encode histone H3 protein. It's not that there are only one or two genes so that you can easily mutate. There are many genes. So all these study I showed, they are based on the indirect inferences. So you can say that, okay, acetylation increase happen in one cancer, decrease in happen, but directly, until and unless you make the mutation, you cannot say with the 100% confidence. And to make the mutation is a very, very difficult test. But nowadays with the CRISPR, so that is where our lab is working. We are trying to understand these questions by mutating directly in the cell line. Then only you can say that, okay, K20, if in the cell line, if you make the mutation K18, then you can say that, okay, this. Otherwise, there will be always some indirect effect. Because of some other reason, K acetylation is high. Right? You can always say. So that is one. So this is the basically the timeline in epigenetics therapy. And the point I am trying to make that these are the years where the base foundation has been done. And these are the things like in 2024 also FDA approved HDAC drugs have been there. 2020 FDA has approved azacitidine, which is a DNA methyltransferase. Decitabine, you have heard this name. You Google it, AI will tell you all these things. Okay. So this is the timeline now from 1942 to different time, different type of drug. But all this has a problem because they had the pan target. Still, specificity could be a question seeing the such a complex biology. This is also Messi's slide, just point you to show you that in glioblastoma, since I have worked some experience in postdoc in glioblastoma, so you basically classify the tumor best, and then in that classification, now you include the epigenetic modifier also. And so whenever you do the genome sequencing experiment, you classify everything. This is now possible that you find out mutation important for modifier, you find out mutation important for oncogene, tumor suppressor gene, everything you can get nowadays. And then this is where the first time mutation has been discovered in the histone H3. And that is where my postdoctoral work was there. So this is the first report back in 2012. So two group, one from the St. Jude, another from the Nature. They first time reported that actually histone H3. So the mutation in the substrate itself is there. Like K27M, K27 trimethylation as example, I am giving you so many times. I give you K36. So G34 has been where mutation is there, and also K27. And a lot of like 2012 to now 2025, the field has been so much expanded that it's a lot of things has been done. So the landmark is this is where and glioblastoma is very difficult. Pediatric high-grade glioma is a very rare tumor, but it is very difficult because no chemotherapy is available. Recently, FDA has approved one therapy. So epigenetic drugs has been used for many targeted, this is just a textbook knowledge. People have done for angiogenesis, people have now area is increasing where you can increase the immune infiltration by treating the epigenetic drug. These are the other examples people have treated with this. I'm skipping fast this slide because I hope you understood now, right? That's why I'm skipping it fast. I'm not skipping it fast because I need to finish the lecture. I'm skipping it fast because I teach everything. I teach you the basic that why chromatin modifier is important. I'm telling you that you can target cell cycle, you can target angiogenesis, you can target metastasis. So if you know the specific enzyme, and these are the literature is available, you can read it. Okay. So the modern day cancer epigenetics is classify the tumor, go to the epigenetic modifier, design the drug, and test it. So this is how people have come up with this. These are the signaling molecules. Again, this is not the for the talk, because signaling molecule has been the time when people are using the kinases for the as a cancer treatment. Now people are combining with the epigenetic modifier, and the one I talk about, K27M and G34R. So this is playing an important role apart from the other mutation. Also, you can design EGH2 is the H3K27 trimethylation inhibitor. So these are the proof of concept. People have done it, and now in your tumor classification, you can involve, apart from copy number transcription, you can also involve G34 mutation and K27M mutations. Okay. So you could target intrinsically, or you could target the tumor microenvironment. And so in epigenetic factors, you look for the genes which could target this DNA damage as well as the chromatin modifiers together, or you can target combined immune checkpoint inhibitor with the chromatin inhibitors. So combination therapy you can always come up, and your model system could be any of thing. You can use the organoid model, or you can use for the heterochromatin. I showed you the tumor cancer model right in the beginning of the slides. So this is the way I also discuss about, if you want to prove the nucleosome, since you have read this and you know, because it's the last class, so this is one way you can doing. So this is the actual experiment where it has been shown that indeed nucleosome remodeling is important, and I think this can happen through different way. I told you also. And what is important point is the nucleosome remodeler has also been involved in the many cancer types. If you read about the many groups paper, people will you can able to understand. So I covered mostly the biochemistry of like histone H3 and H4, but these are the one which is actually chromatin remodeler, which I have not covered in detail. But chromatin remodeler, the one which is highly mutated, and they have different role in different disease parameters. So apart from H3, H4 chromatin modifier is a reader, focus on the chromatin remodeler also is there. Okay. And these two groups is working very nicely, because chromatin remodeler has been 20% it is mutated. So apart from P53, so when you talk about cancer, P53 is the highest, and then the more highest is your chromatin remodeler. Okay. You can probe the modifications like this, normal cell, cancer cell. These are all I have discussed, and these are the name of the inhibitors which you go for this. So here I'll spend some time. Okay. So people have approved this DNMT methyltransferase inhibitor. This one has been approved. You people try in combinations, and also the HDAC inhibitor, the different names you might have heard. And the bromodomain inhibitor protein is basically JQ1, which is important for BRD4. So these are all well established FDA approved chemical inhibitors. This target DNA methyltransferases, this target the HDACs and the bromodomain inhibitors. So all clear, right? Name, even I don't remember name right now. So for different tumor type, you have a different inhibitors which has been cataloged, either they are in the clinical trials or they in the So if you summarize, you have basically the DNMTs, 5-azacitidine, which is used for to inhibit DNA. SIRT1 is acetyl inhibitor. So you can use the inhibitor against nicotinamide. So nicotinamide is the cofactor. So you can basically use the some cofactor which is used. For long non-coding RNA, also people have designed some inhibitors, and then you have also for this hepatocellular. So all type of cancer people have designed inhibitors, and these are the few inhibitors which has been approved. So this paper you will find all the details. Azacitidine, which is a DNMT inhibitor, and it can be administered either intravenous, subcutaneous. Some are not approved yet, some are in the clinical trial, some board specific approval is there. So the patient which are in the terminal stages, sometime board approved that any therapy is not working, there are some indication, let's give them. So this is what it means. There is still a lot of adverse side effect for every drug you will use here. And the reason I explained you, right? Now you convince, right, that why this adverse effect is there, because they are very general targeting. So someday, if you are able to come up with the things, the same thing is there here. This is just copied from the same paper. All the drug has adverse effect, but this adverse effect can be treated. So FDA has approved. For the cancer patient, even if you increase the 5 days, 10 days lifespan, it is a huge deal. FDA approved. If your drug can make a person survive more than 10 days, 20 days, 5 days, less side effect, you don't need to cure it 100%. Same deal, AGH2 inhibitor is the one, K27 trimethylation is in the leading. Okay, so like any other drugs, we'll have a drug resistance also. So within 1 and 2 minutes, I will finish it talking about drug resistance, and we will be done with the lecture, okay? So like any drug, you will have a different resistance mechanism. So these drugs also resistance mechanism could occur if you are using the epigenetic drug. You know about all the other type of drugs. So these are all the chemotherapy. So the resistance will develop some point or other point. And the reason to develop resistance are many. Sometimes they change the DNA methylation profile. They can change the histone acetylation methylation status. They change the non-coding RNA profile. They can change the methylation status at the genes important for the cancer pathway. It could be oncogene or tumor suppressor. So the cancer could evolve because they could come with the resistance mechanism. And these are the one which has been discussed in this particular paper, very extensive review. And I put this slide for histone demethylase because this is where we were trying to understand. And